Dear participants of OHBM 2020, I would like to welcome everyone to this Taorad webinar during the annual OHBM conference. During this webinar, we will give you some deeper insights into the technology and applications of polarized light microscopy, also known as PLI imaging. In this context, I'm pleased to welcome Dr. Markus Axer from the German Forschungszentrum Jülich. Markus heads there the fiber architecture group and uses and develops the PLI technology now since several years. At the Forschungszentrum, Markus uses a microscope from Taorad now for over 10 years, which has ever since provided services in a maintenance-free manner and without any technical problems. Therefore, at the end of the webinar, I will give you some more insights into the services and products of the Taorad GmbH. And now I will hand over the floor to Markus, who will give you some deeper insights into PLI technology. Markus, therefore now the audience is yours.
Yep.
Okay.
All right. Hello, everybody, also from my side. My name is Markus Axer, and I work now since nearly 15 years in Jülich at the research center, focusing on polarized light imaging using polarimeters and generating images, as you can see here on the front. This is, by the way, an image of a cerebellum of a vervet monkey brain, and you can very nicely see the different scales you can address this technology. And my part today is to give you an idea on how it works, what you can do with polarized light imaging, and what are the physical principles behind this. So from our side in Jülich, a lot of things started with Brodmann, who was one of the first, I would call him already a modern mapping scientist. And he's much more known for his cytoarchitectonic work. But in fact, in 1903, he wrote in one very short report the following sentence: "I stumbled upon the idea of formalin-fixed tissue much later and realized with satisfaction that formalin fixation does not impair the birefringence of myelinated nerve fibers. Therefore, we can study nerve fibers hardened and conserved in formalin with polarization microscopy and observe possible pathological alterations." And this is exactly what we're doing in polarized light imaging. We utilize the what we call birefringence of the tissue. The birefringence is mainly caused by the myelin wrapped around the axons, and we can determine the orientation of this myelin in the tissue, which is, of course, along many axons. So what we can extract then is the birefringence, the orientation of the myelin, the orientation of the entire fibers. And we use polarization microscopy for this. Over the years, we have started to implement entire pipelines, workflows to deal with the data, to deal with a preparation. And one thing is sure, you need to know how to prepare a brain according to the needs of polarized light imaging. So what you can see here on the slide is our general idea of what we do in the institute. We start with postmortem brain, do the sectioning, do the measurement, and then analyze the measurements section-wise. And afterwards, we try to do a 3D reconstruction to get back to the original brain shape if possible. And along this road, we had a couple of publications extending and developing the methods, but also for different applications. And you can see today a few of them in my presentation. In principle, we call it, what we do, 3D polarized light imaging, and it is always, in my view, a combination of the microscopy plus the analysis, because polarization microscopy requires a post-processing of the images you've taken. It's not like most of you might know from bright field microscopy and stained sections, that you directly can use the images. If you want to extract the orientation of fibers, you don't see them immediately. You have to calculate them. And I will give you a flavor on how this works. Everything starts with the preparation, as I said. You have to do cryosectioning, because it's important to keep the myelin untouched, which is not the case in paraffin embedding. So what we do is we use formalin fixed brain, put it in glycerol for protecting the tissue from having some freezing artifacts, and then we section the brain at about 60 micron thickness. What we always also do is, during the sectioning, we obtain so-called block phase images. So after every single sectioning step, we make a photograph of the frozen block, right? And what you can also see is, if you take the section away, every section is deformed non-linearly and in a unique way. This gives us the most headaches at the very end, to really reconstruct the sections from these reconstruct the volumes from the serial sections. And for this, we use these block phase images, and you can see on the right bottom side, you can see a 3D reconstructed block phase image volume, which we use in addition to MRI, prior MRI before sectioning, to realign our sections properly. In Jülich, we are using two different types of microscopes. One is not a real microscope, but we call it large area polarimeter, which allows to measure one large section with one image. But with this setup, we can only reach in plane resolutions of about 60 micron or 64 micron. And with the other device coming from Kurt Hoffmann's Taorad, LMP3, as already mentioned, we can address 1.3 micron in plane pixel sizes, which means that we can address individual fibers in regions where the fiber density is not too high, given the thickness of the of each section. But you also have to deal with the big data, right? So an entire large scale human brain section results in about 30 gigabyte of data. I can show you a video on of this large area polarimeter. So I place the section in between optical filters. I show it in the next slide, and then we rotate the filters, and we can see dramatic changes of light transmission through the tissue, and this light transmission depends on the orientation of the fibers. And here you can see a movie built out of individual measurements we did in one coronal human brain section. And you see on the left hand side the general setup, which is composed of different optical filters which can rotate with respect to the fixed tissue. And on the right-hand side, you see the changes in intensity, and you can see it even by eye if you rotate the filters around the unstained section. So again, we don't use any kind of stain. We just use the intrinsic properties of birefringence. And what you can also see is, depending on where you are in the brain, you always measure sinusoidal curves, but with different amplitudes and with different phases. And this is completely related to the myelination density, but also with the orientation of the fibers within the section and out of the section. And whenever we've done a measurement, which means we acquire images every 10 rotation degrees over a window of 180 degrees, we can then fit the different intensities for each image pixel and extract 3 mathematical parameters, which are the mean value, the phase, and the amplitude. And we call the mean value transmittance, the phase, the direction, and the amplitude the retardation. And you can see the results on the in the top row of this on this slide, and you can also see that it provides 3 different views of the same piece of tissue. And the transmittance, for example, looks a little bit like a myelin stained section, which is not completely true, but it is close to that. The main question is, when you do the measurement and the fitting, it's just a mathematical procedure. But what are the real neuroanatomical correlates here, right? And if you have a closer look to the physics behind and describe the quite simple optical path, you can see that the measured intensity depends on these 3 parameters we extracted from the fitting. And these 3 parameters can be used to determine a 3D orientation vector of the general birefringence. And completely equivalent to MRI, diffusion MRI imaging and visualization, we use a color coding for each orientation vector extracted. And this leads to the so-called fiber orientation maps. You can see here 2 examples at different, measured at different resolutions. And you can see, in particular in the left image, showing the visual system in the vervet monkey brain. You see that we do not only see white matter fiber tracts, we also see their entrance into the cortical regions. And we can also see the differences in the architecture, even in the cortex. And that's one of the main benefits, from my point of view, of polarized light imaging. You can, without any staining, you can generate contrast between fiber structures, but you can also try to give them an orientation. And this mixture gives you a really nice idea on the changes of orientations of fibers. So what can you see at what levels? And here are just a few examples. I always call them knitting patterns. So you can address larger kinds of fascicles. You can see really how they change their sizes, how they fan out. You can go for U-fiber systems in the connecting neighboring gyri. And you can even go for cortical fibers wherever the density of the fibers is not too huge. Right? That means basically in the cortical region. So in Jülich, we have addressed a couple of species, all done with the same type of measurement, from the tiny zebrafish to the pigeon brain, monkey, mouse, rat, human. It's all addressable with the same resolution, with the same quality. However, of course, if you want to go for an entire human brain, that means you have to measure something like 3,000 sections, which easily adds up to a few petabyte of raw data without having done anything on the analytical side. So that is something which gives us a bit of headache since a couple of years, but utilizing high performance computing workflows and also data management tools, it's possible to tackle these problems. So I just want to show you two examples what we did in the past on the data. So one thing is, of course, everybody wants to relate PLI to diffusion MRI, and sometimes it's not beneficial to have this very high resolution, but to downscale without losing too much information, and also aligning the PLI with the diffusion MRI at the same scales and with the same type of data structures. And for this, we had developed some statistical analysis on the high resolution orientations to generate orientation tensors similar to diffusion tensors, and even distribution functions. So with this, we can rescale our data quite easily and then directly compare to diffusion MRI, and that seems to work quite nicely. However, maybe just going one slide back. However, it's not the same, really the same type of data structure we extract, because of the different characteristics of the information we extract as compared to diffusion MRI. So one-to-one comparison is not that straightforward as it looks like. As a last example, I would like to show also how it might look like going into 3D space. So really aligning the individual sections back again into the 3D space. And we did this here in example of 230 vervet monkey brain sections measured at highest resolution. And the movie shows you just the amplitude of the measured birefringent signal, 3D reconstructed. There's no tractography or something, and it's a rendered volume of these amplitudes, and we had to reduce a little bit some contrast, and we had to see something. And what you can really see are already the individual tiny fiber bundles in the different brain regions. And in near future, very near future, we will go for an entire human brain with this technology, with the Human Brain Project, with this European project. And we will also share this in the context of the Highball Consortium. Some of you might know, coming from Canada. So we have an international lab last week officially introduced to the public, and in this context we will also bring all these data sets into one reference frame, into one high resolution atlas, and combine all virtues of the different histology approaches. And this is really fantastic possibility to do it. So if you're still interested in some very specific work, we have 2 posters here this year at the OHBM, so please have a look. I think the links of the posters were also sent with a advertising email a few days ago from Kurt Hoffmann. So have a look, and if you're interested, just contact us. So that was my presentation, and of course we are now open to discuss with you. If you have any question, feel free to ask now.
Thank you for the talk. That was very interesting. I had a few questions regarding the thickness, and if you are able to measure the actual component of the fiber orientation. Because I'm working with a similar kind of setup, but we are using block face histology instead of thin slices, and we are using OCT, and we were thinking of maybe using polarized OCT to capture the 3D orientation and also the actual component. And is your setup able to see this actual component, or only the lateral orientation within the slice?
Yes, so that's a very good question. So we can see the actual components best with the new microscope which is being developed with Taorad. So what I haven't told you is that connecting to Kurt's short presentation is that the PLI, as we have used and developed it over the years, is a flat measurement. So there we have the possibility to determine also the axial orientation, but with a couple of uncertainties. And that was a trigger for us to collaborate with Taorad company, saying we would like to have measurements also from oblique views. So that means with the new microscopes, we will be able to extract the in-plane and the out-of-plane, or the axial and the lateral orientation of the fibers with much more precision as we can do now. So it is possible, but only with this opaque kind of measurement.
Thank you.
And I think, yeah,
sorry. I was just going to say,
OCT is a similar principle, right? But the physics behind is a bit more complex, because you enter the tissue in from a different view, and it leaves the tissue again on a different angle. So calculating, this is one reason why this actual component is even more difficult to extract in OCT currently.
Yeah, yeah.
I was just going to ask, following up on that, if like in the work that you've done without the 3D PLI, like how accurate you think the axial component that you're extracting is? You think it's just a sort of a guesstimate, or is it kind of in the ballpark of where it should be?
So it is. It really it really depends on the brain region and the complexity of the underlying arrangements. And we've seen now that when we use the opaque measurements, we can also quantify this uncertainty pretty well. So there's we presented a solution. One of the posters is dedicated to exactly doing this. So I think in former times we were not really sure how it worked, but with the tilting and with the additional information we can use, we can really come up also with quantitative values. And there you can also see how trustable the different things are. And in PLI, it is the case, even with using oblique measurements, that you have the strongest bias in 90-degree out of sectioning plan, even with the tilting.
Thanks.
And this is, so I mean, clearly there are, in addition to hardware solutions, we are currently trying to learn from simulations how to interpret these critical regions in a quite robust way. But there's nothing published yet, but we are working also on this. So the combination of new hardware plus new technology, software technologies, I think it will really help us to be much better even in these complex zones.
Okay, thank you very much. So maybe I think it might be a good moment to give you some insights on the new microscope from Taorad, as they were just mentioned. So as Marcus said, the first generation of microscope realized PLI in a classical approach, as described already in many publications, a kind of flat measurement, as Marcus just mentioned. Now the new generation of microscopes brings in an additional feature, which is oblique illumination, and which addresses the issue of the fiber inclination or component, as you just mentioned and discussed before. Now the first slide here shows you some of the first generation microscopes, namely the MP1 on the left side, which was the first microscope developed by Taorad, and in the middle the LMP1, L stands for large, which was developed in order to analyze also samples like whole human brain slices, which are larger than the standard SPS format. Now the right image shows Marcus in front of the LMP1, and you see the dimension of a slice of a human brain. Now I must mention, and we are proud about it, that the LMP1 is now operational at the Forschungszentrum for 10 years, and this in a maintenance and trouble-free manner. This is also a strong point of Taorad, since we are only using high-quality parts of well-approved partners. Okay, and here on the next slide shows on the left side a sketch of the new microscope LMP3D, which can deliver the mentioned oblique illumination. In the shown configuration here, the microscope is also automated regarding sample loading, which will allow 24/7 measurements, in example overnight or during weekends. In this automated configuration, we name it LMP3DA. Microscope with oblique illumination and without automation are named LMP3D. And without oblique illumination, we name them LMP2. The 2 stands for second generation of large microscopes for brain imaging. Now it would be possible to upgrade from the LMP2 to a LMP3D or to a LMP2A or LMP3DA. In addition to these microscopes, we also provide a smaller version of microscope, the MP2, which is the initial MP1 in its second generation. Now 2 of the LMP3D will be delivered to the Forschungszentrum within the next weeks, and the microscopes are therefore available from now on for the community of brain researchers. Okay, now Taorad not only builds hardware but also provides software for the data acquisition, which was designed according to the workflows within a lab analyzing many thousands of brain slices. And in the case you will analyze a whole human brain, you would be confronted to around 3,000 of such slices. In this respect, we designed the user interface accordingly and also use, after consultation with the Forschungszentrum, the data container format HDF5. So I think I will stop my presentation at this point, and in the case you have any questions regarding hard and software from Taorad, feel free to send us an email to info@taorad.com. So in the case further questions exist, please feel free to ask.
I had a question. Maybe you mentioned it, but sorry, it's a bit late over here, so it's hard to keep the focus. the images you presented are mainly from healthy subjects, but what happens when you go into pathological brains? And here I see two categories. It would be like Parkinson's, Alzheimer's, so it's something that is not so focal. But what about, well, that's because I'm working with disease like Alzheimer and Parkinson's, but I have colleagues working with multiple sclerosis and glioblastoma. So knowing, and we are an MR center, so knowing what's happening at the fiber level would be supremely interesting. So,
yeah. Yeah. So of course, I'm coming from a mapping institute, so that's the reason why we always start with normal brains. But we have some experience now with multi-system atrophy brains, and what you can see in case of degeneration is that you have completely different signal strength available. So this technology is really sensitive to myelin, which means if you have a degeneration of myelin, you can immediately see it and localize it properly. But you still see fibers if they are still present.
Yeah, you have the information of orientation, and then the other one is sort of the, yeah, you had those three parameters like the intensity, or I can't remember exactly the.
Yeah, exactly. And in this amplitude, there is a part which describes the myelin components. So with this, you're really sensitive to MS. I'm pretty sure you will see it, but always in comparison with the normal brain.
Yeah. Okay. Yeah. Linking this with techniques like quantitative MRI, where we measure the quantitative MR properties of the tissues, but with of course the resolution of MR. So it's submillimetric at 7T, but not down to what you reach. Linking fiber and MR property would be very, very, very nice and informative in both ways, in making sense of what we see at the MR level and what you see at the larger scale.
I completely agree. So we have currently a running grant in the Human Brain Project together with colleagues from Neurospin in Paris.
Yeah.
And with them, we did an entire human hippocampus reconstruction, first with diffusion, and then with PLI. And on top of that, we also do two-photon microscopy inside the sections, so that we have this three different components and different scales available. And we all have to learn what we can expect from the different measurement signals. And, especially concerning this micro structural information, this is not straightforward.
Yeah, and it would be a way of validating some stuff that the MR physicists are pushing, and if it corresponds to something real in terms of the microstructure, then it would be nice, because then we know what we're talking about. And yeah. Okay.
Yeah.
Thanks.
You're welcome.
Can you still hear me?
Yes.
Okay. I had a couple of quick sort of, I guess, more practical questions. Thank you very much again. I really appreciate that you guys are talking about this and pushing this. I think it's amazing stuff, and it's nice to see that the microscopes are almost ready to be released. So that's amazing. I had a couple of questions, one around tissue warming. So obviously acquiring a whole slice of a human brain is going to take a considerable amount of time. Have you run into issues with warming of the tissue and that affecting the signal, or?
You mean warming up the tissue?
Yeah.
Yeah. So in principle, we do all our measurements at room temperature.
Mm-hmm.
What we observed over the years is that if you cut a frozen brain and then bring it into room temperature, and then also put it into some kind of liquid, crystals start to move around, to diffuse across the sections. And it is mainly cholesterol which is forming crystals. And this means that you have to do your measurement very quickly after your brain preparation the mounting of the sections. So usually we do our measurements 2 days after the complete mounting of everything. So you really have to do some logistics on this. And forming crystals is always a problem for PLI, because PLI is sensitive to many of the crystals. Not all crystals generate birefringence, but some of them. And you start to see them on top of your sections, and you don't want to have them there.
Okay. And then I guess just to touch a little bit on the processing pipeline that you guys have been able to implement in your lab and at Jülich. Like obviously you have access to some nice supercomputers and such there. I just wonder how practical you think it is for, you know, a relatively small lab or a group of labs to get together and try to put together the same type of or a similar type of infrastructure that can process these images in a reasonable amount of time, I guess. I mean, it all depends on the size of obviously on the size of your slices, but like, let's say you wanted to do a hemisphere or something like this.
Yeah, I think it's still possible. It just takes much more time. If you want to do fitting of every individual pixel, it takes some time. If you have an individual cluster, it is possible, but it takes just a bit longer, or let's say a couple of weeks longer, I would say. But it is doable. So we deal with HDF5 file formats, and they are also usable in smaller kind of clusters, and you can even use there the high performance computing methods.
What's the scale of things that we're talking about? I remember the raw data like 30 gigabytes, but then the reconstruction, you say couple of more weeks means the original processing is already weeks long.
So the raw data is even more. The raw data of one entire human brain, a large human brain section, is something like 800 gigabytes. And the 30 gigabyte are the condensed and analyzed results. So if you really want to go for 3,000 sections, it's about a few petabyte of data you have to deal with.
That's impressive.
That's a nightmare.
That's another way of putting it.
Yeah,
that's science.
Yeah, but you know, there will be much more also computing resources available through the Human Brain Project, for example. So if there's interest for collaboration and sharing data, in the end, you can also ask for support in doing supercomputing and also using supercomputing resources. Every lab can do this worldwide. So I think the future will bring much more possibilities like that.
Yeah, I guess, well, these kind of resources, just the storage and the processing power are not just, you can't just go buy it at MediaMarkt and get that running under your desk. So it calls for more global infrastructure.
Yes.
That makes sense. if I show up in at home and say, well, I need 10 petabytes of disk space, that will not go down very easily with them.
Yeah, that's true. That's true.
Thank you very much for all the information. Cheers.
Thank you.
Cheers. Thank you for joining.
And thank you for doing it so early / late.
Okay. Okay, thank you. Bye-bye.
Bye-bye.
Bye-bye.